Hello everyone, welcome back to the channel. So today we're going to be talking about mutations and specifically the evolution of the mutation rate. Mutations are one of the fundamental forces of evolution. They are obviously what provide variation to populations, without which evolution just couldn't happen at all. And so it's a very important component of evolutionary biology, and it's something that we've generally been interested in for a century and a half now. Not only just the mechanism of mutation itself, but we're also interested in the rate at which new mutations are added to the population every generation. And one of the cool things that we've seen is that across the tree of life, there exists an order of magnitude change in the mutation rate. So, one of the big questions is why is the mutation rate so variable across the tree of life? And there are sort of three broad answers to this question. One is that there exists differences in DNA repair mechanisms. So some organisms may have DNA repair enzymes that are just super efficient and that can weed out the vast majority of mistakes during replication. Other organisms may have much less efficient or completely lack DNA repair mechanisms, and so therefore just have more mutations because of that lack of a way to repair mistakes during replication. Secondly, there exist differences in the strength of selection across organisms. So I'm showing you this plot from this study in 2017. On the x-axis is population density, on the y is the mutation rate. They've just plotted a bunch of different critters across the tree of life, and what you can see is a pretty strong negative correlation between the mutation rate and the population density. If you've watched any number of videos on this channel, then this should jump out at you as being indicative of the strength of selection. So remember, selection and drift are kind of always battling it out. And who wins is determined by how big your population is. So the bigger the population is, the stronger selection is, because selection is able to overcome the stochastic fluctuations in frequency every single generation that's caused by genetic drift. If mutations are in general bad, then what you would expect is that selection should be trying to keep the mutation rate low. Therefore, if selection and drift are battling it out, then big populations, selection should win. We should see a low mutation rate. In small populations, drift should win. We should see a high mutation rate. And that's exactly what we see here in this negative correlation. Third is that there exist differences in life history strategies. So some organisms may have virtually one or two cell divisions before they begin to reproduce, whereas others may have many, many cell divisions before they're able to reproduce. And what we mean here is that the amount of time it takes from birth until you reach sexual maturity and can begin to reproduce, that expanse of time dictates how many cell divisions there has happened. And so the more cell divisions, the more possibilities there is for error during replication, right? So these three things kind of broadly represent why the mutation rate is so variable across the tree of life. And we're going to dig into each one of these in more detail as we progress through the video. So the main point of this video is I was really excited about this specific paper that just came out a few days ago in the journal Nature, titled "The Evolution of the Germline Mutation Rate Across Vertebrates." This paper is really, really cool. My Twitter feed has been abuzz with it. And we actually read it at my lab meeting this past week. We presented this paper, and so I thought it was worth it to sort of share this paper with you so that you can get an idea of why it's cool and some of the really neat things that they were able to find in it. So what did they do? Let's look at the broad things that they did. So first, they used trios to measure the germline mutation rate. So trios are when you have the genetic sequence from the father, the mother, and the offspring. When you have all three, you can get a direct measure from the pedigree of how many mutations have occurred from parent to offspring. So a direct measure of the germline mutation rate. That's normally really limited to a handful of model organisms we can pull that off in. So that they were able to do this in 63 species of vertebrates, including mammals, reptiles, birds, and fish, is pretty cool. That's a pretty exciting thing that represents a taxonomic scale. We have not been able to do this in the past. So with this gigantic data set, they were able to ask questions like, what were the most common types of point mutations? Point mutations being just simple base pair changes from, say, an A to a T or a C to a G, right? Just like simple base pair changes, that's a point mutation. They looked at what were the most common types, were there differences across different taxa for which one of those was the most common, et cetera. They also looked at their mutation rate estimation in relation to different life history things. So, like I was talking about before, like the average age of the parent at reproduction, what is your generation time, those sorts of things. They also were able to compare their mutation rates across measures of population size. So, looking at the impact of selection, just like we were talking about. And then finally, they also looked at the differences in mutation rates between domestic organisms and wild animals. So that was a kind of neat thing that they were also able to do. So let's dig into this paper. We're going to I'm going to show you a bunch of figures. We're going to talk through their results, and then I'm going to try to help you sort of understand what each one of their results sort of indicates, sort of suggests. Okay, so this is their first figure. It's sort of the big, broad take-home figure. On the left is the phylogeny, so the relationship between all the different trios that they had, including fish, mammals, reptiles, birds, like I said before. And then on the right is the mutation rate that they estimated for each one of the taxa. Just looking at this, you can see that there's a relatively tight range with some decent variation around that, right? Humans fall a little bit above this with a 1.17 e to the -8 mutation rate, so that's about 100 new mutations per generation. Most of the organisms here were a little bit lower than that, and some were like a lot higher. So several of the birds and some of the reptiles actually had a much higher rate of mutation than what a lot of the fish and the mammals showed. So that was kind of cool. They also looked at, like I said, what were the most common types of point mutations. So one, they found that there were significant differences across these different vertebrate groups for A to C transversions, as well as C to A transversions. Where mammals had generally lower rates than reptiles did. So that was one interesting pattern. But what they found that was common across all of these groups is that C to T transitions were far more common than any other. And in fact, represented almost 50% of all possible mutations were C turning to a T. So why is this? Why is that change the most common? Furthermore, you'll note here that they have a darkened bar and then sort of like a grayed out or a transparent bar. That represents the dark is the non-CPG sites and the transparent are the CPG sites. So what is a CPG site? As you can see, obviously it's very relevant to why this was the most common one. So let's talk through what are CPG sites and why do they mutate far more often than any other site? Okay, so let's start in this top corner with this little thing here labeled CPG island. So what CPGs are, what that actually stands for is the cytosine phosphate guanine. That's what that stands for. So you have a C, G, C, G as the 5' to 3' top strand, and then going the other way, you have a C, G, C, G, 5', 3' going the other direction. Okay, so that's what those CPG sites represent. Now, what's really cool about these sites is that they are very often targeted by DNA methyltransferase. And what DNA methyltransferase is it is an enzyme that binds to CG, to the CPG sites, and it methylates them. Okay, so, and when it methylates them, it actually converts the cytosine to 5-methylcytosine. And what this does is it effectively silences this region. So what you find in a lot of vertebrates is around 70% of the promoters for protein coding genes in vertebrates have CPG sites around them. What that means is that they can be methylated, and that allows for controlling the expression of that gene by methylating these CPG sites. Okay, so that's the first thing to kind of understand about these CPG sites. Is that they can be methylated. However, one of the side effects of methylating cytosine and turning it into 5-methylcytosine is that it's very easy for 5-methylcytosine to undergo what we call deamination. And when it undergoes deamination, it gets rid of that methyl site. It just expels the methyl site. And when you do that, you transform the cytosine into a thymine. So 5-methylcytosine, when it becomes deaminated, is a thymine. Okay, so that's going from a C to a T. So now when you've transformed a C to a T, and then you have your mismatch repair proteins scan along, they find this mismatch because now you have a T and a G site that don't match, that's a non-canonical binding, they will then repair it. And what they tend to repair it to is not back to the CG, they tend to repair it to an A and a T. Okay, and so that's what I'm showing here. The mismatch repair protein changes then the G to an A, and then now you have an AT pair. Okay, so what this indicates is that CpG sites first are very often targeted for methylation. However, when they are methylated, the 5-methylcytosine very easily transforms into a thymine whenever it undergoes deamination. And then when mismatch repair enzymes come along, they tend to not change it back to the CpG, but instead change it to an A and a T. I know that was a lot of information. Hopefully you kind of followed the general logic there, but this leads to some pretty interesting patterns in especially vertebrate genomes. So if you imagine some ancestral genome, shown here, where each one of these little dashed bars represent CpG sites. Then over time, what happens is these little yellow circles, these are the methylated CpG sites. So those sites tend to be, targeted for methylation. But since they happen to mutate a lot more readily to AT sites, then what over evolutionary time you tend to find is that CpG islands end up being the only thing that's left. You have lots of A and T sites that used to be CpG sites, but they've been mutated. And so you find very few CpG sites across the genome. This is actually represented by the fact that if you just compare the CG content, so the GC content across our genome is like 45, 48%. But the number of CpG sites you can actually estimate from just like the general content alone. And we actually only have about a fifth of the expected number of CpG sites given the GC content in the genome. So that indicates that there's a very, very large mutation rate associated with CpG sites. So this has pretty big implications, for example, the spread of transposable elements and many larger genomes, right? And another thing that tends to happen in larger genomes is that you find a dearth of CpG sites relative to what is expected. So how do we explain this? Well, the idea goes is that ancestrally we had lots of CpG sites. And so transposable elements, they inserted into the genome. And when transposable elements are not silenced, they proliferate and they cause problems, right? So what you initially want to do is silence them. Well, that means you need to methylate the regions near their promoters. And so what would happen is if you get a transposon that inserts where there's a CpG site, then you can methylate it and silence it. But remember, methylating those CpG sites allows it to then mutate away from the CpG to an AT. Right? And so initially, if you have lots of transposons that are being inserted into the genome, they're proliferating, and then all of your CpG sites end up having to get methylated to silence them, what this eventually will lead to is a dearth in the number of CpG sites relative to the GC content because they've all become mutated to A and T. And we find this exact pattern. So in the B panel here is genome size on the X with the total number of transposable elements on the Y. And you can see a very, very strong positive correlation between those two things. Now look at the opposing pattern. Genome size again on the X, but the observed CpG divided by the expected just based on the GC content in the genome, is a strong negative correlation. Again, indicating that as these transposons accumulated, they have taken over the CpG sites. We need to methylate the CpG sites to shut them off. But when you methylate your CpG sites, they then become very easy to get deaminated and mutate to A's and T's. So the argument, the idea around why we have CpG islands, why we have so few CpG sites relative to the GC content, is because of the transposable element proliferation. That was a bit of an aside, but I thought it was a really cool point and worth sharing. So let's get back to the paper. So another one of the things that they found is that the mutation rate is positively correlated with a parental age at reproduction. And you can see here on the x-axis is the average parental age at reproduction. Y-axis is the mutation rate per generation. Very, very strong clear pattern emerges here. It should be noted that the organisms that are largely driving this pattern, however, are mammals and birds, right? So the fish and reptiles are all kind of nestled down here towards the origin. And it's difficult to tell if there's a whole lot of pattern, but this is mostly because fishes and reptiles don't have as much time. So the average age to reproduction is much, much, much shorter than mammals and birds in general. Another thing that they found is that there's a strong male bias in the mutation rate. And so if you think about who's actually contributing to mutations in offspring, across all the organisms they looked at, except for fish, there's a strong male bias. So males are contributing most of the mutations. One, notice here that there's a conspicuous lack of amphibians. There's a few reasons for this. One, because it's very difficult to do trios in amphibians. Two is that amphibians have gigantic genomes. So just getting the sequence data for amphibians is much, much harder than for most of the rest of vertebrates. So there's this lack of amphibians. However, it's important to note that amphibians reproduce a lot like fish, and that females produce lots and lots and lots of eggs, right? And so we're gonna talk about why that matters here in just a moment. So let's think about these two previous plots, that there's a positive correlation between the age of reproduction and the mutation rate, and that there's a male bias in who is contributing, which parent is contributing most of the mutation. So those are the factors. Now let's see if we can conceptualize why exactly that is. So imagine down here we've got our little cartoon. I'm showing them as humans. Any of the vertebrates except for obviously fish work in this context. So blue is for the females, red is for the males, and then on the x-axis is the time to reproduction, and the y-axis is the count of mutations. So the longer and longer it takes before you are able to reproduce, the more cell divisions that have happened. Right. So, and the more times your cell divides, the more times DNA replication has to occur. The more replication occurs, the greater the chance that there are going to be some mutations, right? So that helps to explain why we see this correlation between parental age at reproduction and the per generation mutation rate. Secondly, is we also need to note that that there could be some difference in the ages of the parents themselves. So that you could get, if males, for example, take longer to sexually mature on average than females, then males could be contributing more mutations per generation than females. That's one particular cause of bias, but it's not the one that's the most explanatory. The one that's the most explanatory is the difference in male and female gametes. So male gametes in vertebrates are small and motile, whereas female gametes in vertebrates are large and immotile. So they don't move and they tend to be bigger. Because of this, males tend to produce a lot more gametes than females do. And what this means is that males undergo a lot more cell divisions in their gametes, in their germline, than females tend to undergo. And so because of this, there's a lot more chances for male gametes to accumulate mutations than female gametes. So just even the germline mutation rate from time of birth to the time of reproduction between the sexes is dramatically different, just because of the number of cell divisions that happen per unit time, right? And so this helps to explain why we see this strong male bias in the mutation rate. Okay, so now let's think about why fish and probably, I hypothesize, amphibians wouldn't follow this. So remember, in most fish, they have lots and lots of eggs. Is that the difference between the number of eggs produced versus the number of sperm produced by males, that differential is much smaller in fish and probably amphibians than it is in most of the other vertebrates. And because of this, you don't see this huge discrepancy in the cell divisions between the two. And so I think that's what they propose. And I think that's a really good explanation as to why we don't see this male bias in fish. I suspect we also wouldn't see it in amphibians, but we do see it in the rest of vertebrates. Okay, so now let's move on to looking at their correlation between the population size and the mutation rate. And to understand this, we're going to need to do a little bit of background. So first, we need to talk about the drift barrier hypothesis. If you've seen any number of videos on this channel, you've heard of the drift barrier before, but I really think it's super important, and so I'm going to drill it in your head in at least every other video. So, drift barrier hypothesis. Natural selection should act to reduce the mutation rate. Thus, the mutation rate should be higher in species with smaller effective sizes because selection can overcome drift. This is what we talked about in the very first slide, but just to put it into a more formal definition, drift barrier hypothesis. Now, the next thing that we need to recognize is that the population size through time is not constant. And so whenever we're measuring the population size of different organisms, what we're actually doing is we're averaging over time spans. So different measures of population size are actually querying different expanses of time. So let's see if I can kind of help you understand this sort of weird concept. So in this graph, we have the present down here at the origin, and then backwards into the past, and then the effective size on the y. And so in the present, in the most recent past, population size is small for this little hypothetical example. And then in the past, it was bigger. So we've had a recent population decline, and then it was kind of stable backwards in time. Okay, so now imagine we are asking about the drift barrier hypothesis. We want to know, is there a correlation between mutation rate and population size? Well, it depends on which part of the past we are investigating. So I've drawn lines to represent how far back into the past we're looking. So let's say we're investigating the very recent past. So that's with this red line, with this vertical red dashed line, and then this horizontal one is sort of the average. So we're averaging over that time interval. In that time interval, the effective size is low, right? Now compare if we average over a broader time interval. So we go back to blue here. Now notice that average is quite a bit higher. It's not as high as the sort of stable population in the past, but it is higher than if we just average very near the present. So that's a little bit bigger. And then if we average over long time spans, we get the highest effective size. It's still a little bit lower because it's dragged down by the recent population bottleneck, but it is in general higher. So what does all of this have to do with whether or not there's a relationship between mutation rate and the effective population size? Well, importantly, we need to ask, what is the time frame that is relevant to mutation rate evolution? So if we ask, so if we're only querying the part of the tree that is very recently, the part of the population past that is very recent, that might not actually be what's relevant to the evolution of the mutation rate. Remember, mutation rates are generally very slow. And so to evolve, it's going to take them a while to evolve. And even if the population size is dramatically changed, the rate itself is going to lag behind that change, right? So what's probably the time frame of relevance is much broader than the time frame of the very recent past, right? So in this way, what we would expect is if we're querying just very recent, then we might get no correlation, but if we query across long time spans, then we might find a correlation. That's just sort of priming you for what I'm fixing to show you, because I suspect otherwise the graphs that I'm fixing to show you may not make a ton of sense. So that's just sort of the background, and then now let's actually look what they found. Okay, so remember, I'm keeping the drift barrier hypothesis up here, and then here are two different plots. So for one, if we just look within the first 30,000 years of a species history. Okay, so from the present day, going back 30,000 years, what's the average population size over that time? Is there a relationship? No, there's no relationship. You can see right here that across all of these organisms, there's no clear correlation. If we do the same thing, but we look just between 30,000 and 130,000, so we're discarding the earlier part, we're discarding anything deeper, we're just looking at that window of time. And again, it's closer to a correlation. You can see the P is now 0.14. That's a little bit closer to a significant correlation, but it's still not there, right? There's still no clear correlation that is present. However, they do note in their paper that if you just look at mammals, so if you discard birds, fishes, and reptiles, there is a significant correlation in the 30,000 to 130,000. So you can kind of see this if you just look at the dark orange plots or dark orange dots and just imagine removing the rest. Then you can see there actually is what appears to be a negative correlation. Okay, but now let's zoom out even further. Okay, now let's zoom out from 30,000 to 1,000,000 years. right? So let's take a much deeper time span to look at, and that's what they're looking at up here at the top. Okay, so this is from 30,000 to a million. Now we can see across all taxa a very strong negative correlation between the population size and the mutation rate. Right. So again, what this indicates is that the mutation rate tends to evolve at much bigger timescales. So on the timescales of a million years, as opposed to on the timescales of 100,000 years. So that's not enough time for there to reach sort of this, for selection to be driving the mutation rate. It takes a much broader time span. So this was a really interesting component that I hadn't seen done in a lot of studies. Often they will just take one average and they will just say, this is the average, it either correlates or it doesn't. But by kind of chopping up the time slices like this, it not only does it show us where the correlation is, it shows us which time spans are relevant to the evolution of the mutation rate. So I thought that was really cool, and I hopefully that sort of makes sense to you. So the next thing that we might be interested in, now that we have some estimates of germline mutation rates, is we might be interested in what's called the yearly mutation. So the yearly mutation rate is just the germline mutation rate divided by the parental age at reproduction. And the reason that we do this is because lots of organisms have different generation times, right? They may be a lot older when they reproduce. And so by doing this, we can kind of standardize the number of mutations that accumulate in each lineage per year. And so by doing that, we can then compare the yearly mutation rate on the y-axis here with the substitution rate on the x-axis. Remember that the substitution rate is different than the mutation rate. The substitution rate are fixed differences between organisms, okay? And so in this way, we're thinking about in a phylogenetic context as opposed to a population level context. At mutation drift equilibrium, and assuming that there is no selection, we can actually directly estimate what we expect the substitution rate to be as k, which is equal to the standing level of variation, which is 2 times the population size times mu, mu being the mutation rate, multiplied by the rate of drift, which is 1 over 2 NE. Notice that the NE's here cancel, and we're left with just the mutation rate. So at mutation drift equilibrium, and assuming no selection, the substitution rate should be equal to the per generation mutation rate. What this means is that we would expect a 1-to-1 correlation between the substitution rate on the X and the yearly mutation rate on the Y. And we see a significant positive correlation between those two things, with most of the dots falling around that 1-to-1 line, the 1-to-1 being the dotted line. This is a pretty good indication that molecular clocks, even like strict molecular clocks, are probably getting close to the real answer. This is something that a lot of people have been suspicious about for a long time, is how well does the per generation or the yearly mutation rate match the substitution rate. And this study has shown that there's a pretty good match, indicating that the molecular clock assumption is probably not a bad assumption. I want to preface that with saying that most phylogenetic studies don't assume a strict molecular clock. We actually assume relaxed clocks where they can vary per branch. So we very rarely make a strict clock assumption. But if we did, what this study shows is that it's actually not that bad of a fit, right? That you're still getting a pretty decent fit. Some exceptions exist, however. So when we're looking here, I want to point out the reptiles. Way up here, this is obviously this is a very big discrepancy between the mutation rate and the substitution rate, right? So one is that it's generally holds true, but it could be taxa specific. So for reptiles, obviously the mutation rate appears to be way higher than the substitution rate. For mammals, it seems to be pretty close. For fishes and birds, it seems to be pretty close. But for reptiles, we're seeing this dramatic difference. So while there's a general pattern that generally holds true, it can be very clearly taxa specific. And I'm showing you over here on the bottom right, Dickerson, 1971, where what he's plotting on the x-axis is millions of years since divergence based on the fossil record. On the y-axis is the number of substitutions. And I'm just showing you this to look at there's historical precedent for this idea that there should be this sort of 1-to-1 scaling. There should be this very constant scaling through time. That's what they found in the 1960s and 70s. And that's roughly what we're seeing in their data here as well. Okay, lastly, they found initially a significant difference in the mutation rate between domestic organisms and wild organisms. So that's the first box plots here on the bottom left. Um, but there's a very interesting thing that can emerge here. So oftentimes in domestic species, what humans have done is we've bred them to reproduce at younger ages, right? So we make them mature quicker. And so if you remember, the yearly mutation rate is the germline mutation rate divided by the parental age. So you can have the exact same mutation rate, the exact same germline mutation rate, but differences in parental age, and that can lead to a difference in the yearly mutation rate. Right. So what they did is they accounted for this difference in the parental age, assuming that humans have been selecting for younger ages. And when you do that, when you account for that difference, there's actually no difference between whether you're domestic or wild. That the per generation mutation rate or the per yearly mutation rate is actually very, very close to being the same. So this difference that has been pointed out in previous studies and that they found here, it's probably not actually a difference in the mutation rate, but rather reflects selection for shorter generation times in domestic species. Okay, so in summary, they found that the mutation rate is highly variable across organisms. I showed in the very first slide that it's incredibly variable across several orders of magnitude for across the tree of life. For vertebrates, the range is a lot narrower, but there is some variation there. Almost half of all mutations they found are C to T mutations, and that they're occurring at CPG sites. We got into a little bit of a rabbit hole into CPG sites and how CPG sites allowed for the proliferation of transposons, and that's why we actually have much fewer CPG sites in our genome today relative to what we would expect given the GC content. They found that there's a strong correlation between the age of the parent and the mutation rate, and that there's a strong correlation between generation time and the mutation rate. And this is exactly what we would expect just based on the number of germ cell divisions that occur from the time of birth until you reproduce. So the longer and longer that interval is, the more mutations you've accumulated. They also found that there was a strong male bias, right? So for most vertebrates, except for fish, and I hypothesize amphibians, there's a strong male bias in the number of mutations. Again, because males tend to undergo a lot more cell divisions between birth to the time of reproduction compared to females. So that leads to males having more mutations to pass on. Finally, they found that there was a negative correlation in the effective population size and the mutation rate over long time scales, but not under short time scales, importantly. And that supports the drift barrier hypothesis. Lastly, the yearly mutation rates roughly approximate the substitution rate estimated from species divergence. This is really cool and indicates that for most of the time, a simplistic assumption of a strict molecular clock is actually not going to lead to a systematic bias in your estimates of divergence time. So there is a rough correlation between those two things. Okay, so thanks so much for being here. Hopefully you learned something about the evolution of the mutation rate. If you have any questions, feel free to drop them in the comments. I've linked this particular paper in the description box. Check it out. If you read it and have any questions on it, I'll be happy to answer them. Thanks so much for being here, and I'll catch you next time.